Assalamualaikum warahmatullahi wabarakatuh. My name is Nursyafira Asma Humairah and I'm from Sunan Gunung Jati State Islamic University, Bandung. In this occasion, I will do an experiment with the title is The Effect of Visible Light and the Quality Seed on mung bean seed in Germination Process. The X factor of this experiment is the quality of the seed. I add one more factor in this experiment, that is gibberellin hormone, because I want to know this hormone can affect the germination process or not. How do plants know when it's spring and when to grow? If a seed grew too early, it might be exposed to a harsh cold environment. If it grew too late, it might not have enough water to survive. The process by which an organism grows from a seed into a plant is called germination. What triggers the seed to start growing? It varies by plant and by the environmental condition where the plant grows. In this experiment, I will investigate factors that can affect the germination of seedling. Among the various naturally occurring abiotic factors regulating plant development, light or radiations plays an important role in germination process. Solar light consists of electromagnetic radiation with wavelength ranging 400 to 700 nanometer, which categorized to violet, blue, green, yellow, orange, and red color. Gibberellin is plant hormone regulating key processes in plants. Many of them are significant agricultural importance, such as seed germination, root and shoot elongation, flowering, and fruit patterning. So, the purpose of this experiment are 1. To analyze the effect of visible light with different color on the germination process. 2. To analyze the effect of seed quality on the germination process. And 3. To analyze the effect of the gibberellin hormone on the germination process.
The first step to do is, first, prepare tools and materials. The tools and materials that I need in this experiment are cotton, petri dishes, mung beans or Vigna radiata that have been soaked in water for 6 hours, gibberellin hormone with concentration 75 ppm, aqua dust, drop pipette, tweezers, ruler, scissors, thermohygrometer, labels, mica paper with green and red color, and then vernier scale. Next, separate the good quality seed and the poor one. The good seeds are green with no brown and rotten part of it. Next, in each petri dish, put cotton that has been moist with water and 5 mung bean seeds. 4 petris, petri A, B, C, and D for good quality seeds, and 4 others, E, F, G, and H for the poor one. Petri A is a control. Petri B is covered with red mica paper. Petri C is covered with green mica paper. And for the Petri D, add the gibberellin hormone with concentration 75 ppm. Do the same treatment for the poor seeds, Petri E, F, G, and H. All samples are placed in the same place and observed for their development. Don't forget to measure the temperature and the humidity of the chamber with the thermohygrometer. In each observation, measure and record the height of the sprouts.
Observations have been made every 12 hours, resulting in observational data with 12, 24, 36, and 48 hours. The sprout height data taken is the highest sprout per petri, based on the observed data that has been presented in graphical form. The highest germination after 48 hours was in Petri B with good quality seed treatment covered with red mica paper, and the lowest was in Petri H with poor quality seed treatment added with gibberellin hormone. The height difference between seeds covered with red mica and green mica paper for good quality seed was 0.5 cm and 0.9 cm for poor quality seed, whereas when the two treatments were compared with Petri A and E, which were controls, there was a significant difference. Then, the difference between the good quality seeds given with gibberellin hormone and the control, it can be seen that the seeds given with the hormone germinate faster compared to those not given. But in seeds with poor quality, the gibberellin hormone did not affect the speed of germination. This can be caused by internal factors of the seed. Next, the height difference between the good quality seeds Petri A, B, C, and D and the poor quality seeds E, F, G, and H shows that seed quality affects the speed of seed germination. The data shows that germination of mung bean or Vigna radiata L on the red color spectrum is faster than on the green one. That's because the red color spectrum has the most effective wavelength for chlorophyll to carry out the photosynthesis and growth than the green color spectrum. The red color spectrum has a wavelength of 625 until 700 nanometer, while the green color spectrum is 500 until 565 nanometer. In research conducted by Indasari et al. 2018, that the red color spectrum can stimulate faster plant growth. The conclusions from these experiments are: 1. Visible light has an effect on germination and depends on the color spectrum of the light. 2. Visible light with a red color spectrum is ideal for the germination process. 3. Seed quality greatly affect on the germination process, and 4. The gibberellin hormone can also affect the germination process. I think that's all from me. Wassalamualaikum warahmatullahi wabarakatuh.